Hello students, today we are going to study the chapter Biomolecules. Have you ever wondered what are we humans made of? I am sure that carbon, hydrogen, nitrogen, all these elements are coming to your mind. Now if I ask you to find the different organic compounds in a living organism, how will you go about it? We can take a tissue, let us say a vegetable or chicken liver, and grind it along with trichloroacetic acid. We can then filter the slurry using a cotton cloth to obtain two fractions, acid soluble and acid insoluble. These two fractions can then be studied separately. There are various techniques by which we can further extract and purify the individual compounds from the fractions. So, in order to study a living tissue sample and identify a particular organic compound, we make an extract of the sample and then use various separation techniques till we separate a compound from all the other compounds. What we mean to say is that we isolate and purify a compound. We use several analytical techniques that give us an idea of the molecular formula and the probable structure of the compound. Can you now tell me the most abundant element which is found in human body? It is carbon. All the carbon compounds that we get from a living tissue are called as biomolecules. But students, living organisms have also got inorganic elements and compounds in them. How do we know this? To find this out, we perform a slightly different experiment. We weigh a small amount of living tissue, say a leaf or a liver, and this is known as wet weight. We then dry it so that all the water evaporates. The remaining material gives us the dry weight. Now, if the tissue is fully burnt, all the carbon compounds are oxidized to gases like CO2 and water vapor. So, all the carbon compounds are removed. What is remaining is called ash. This ash contains inorganic elements like calcium and magnesium. Today, we are going to discuss the different kinds of organic constituents which are present in a living tissue. These organic compounds or biomolecules are classified as carbohydrates, lipids, proteins and nucleic acids. The most exciting aspect of chemistry deals with isolating thousands of compounds from living organisms and determining their structures and, if possible, synthesizing them. Many of these compounds are involved in various processes of metabolism. Hence, they are known as metabolites. Metabolites are the intermediates and products of metabolism which we are going to study in a while. A primary metabolite is directly involved in normal growth, development and reproduction. For example, carbon, nitrogen, hydrogen and oxygen. A secondary metabolite is not directly involved in these processes but usually has an important ecological function. For example, alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums and spices. Friends, when we had crushed our living tissue in the presence of trichloroacetic acid, we had obtained two fractions, acid soluble and acid insoluble. Compounds in the acid soluble fraction have a weight of 18 to 800 Dalton. These compounds are known as micromolecules, whereas the compounds in the acid insoluble fraction have a higher molecular weight in the range of 10,000 Daltons, and these are known as macromolecules. Macromolecules consist of biomolecules like proteins, nucleic acids, polysaccharides and lipids. Proteins are formed of amino acids. So, amino acids are organic compounds containing an amino group and an acetic group as substituents on the same carbon. This carbon is also known as the alpha carbon. Hence, these amino acids are also called alpha amino acids. Proteins are actually substituted methanes. If we look at the structure of methane, there is a carbon atom to which 4 hydrogens are attached. So, amino acid is a modified methane molecule. There are 4 substituent groups occupying the 4 valency positions of carbon. These groups are hydrogen, carboxyl group, amino group and a variable group which is designated as the R group. Now, based on the nature of R group, there are hundreds of different types of amino acids. However, only 20 of them are found in the genetic code. Since these 20 amino acids are found in proteins, they are called as proteinaceous amino acids. The R group in these amino acids could be a hydrogen, a methyl group, or a hydroxymethyl group, etc. When the R group is the hydrogen, the amino acid is called glycine. When it is a methyl group, the amino acid is called alanine. And when it is a hydroxymethyl group, the amino acid is known as serine. The R group can be a straight chain, or it may be branched, or it may be cyclic. We will take a look at such structures shortly. For example, in proline, the NH2 amino group is replaced by an imino or NH group. Depending on the nature of the R groups, the proteins can be of several types. Neutral amino acids have one amino and one carboxylic group, for example, glycine and alanine. Acidic amino acids have one amino but two carboxylic groups, for example, glutamate and aspartate. Basic amino acids have two amino groups and only one carboxylic group, for example, arginine and lysine. Cysteine and methionine are the sulfur containing amino acids, while serine and threonine have a hydroxyl group, so they are known as alcoholic amino acids. There are certain amino acids like phenylalanine, tyrosine and tryptophan which have a cyclic ring structure. These are known as aromatic amino acids. Cyclic amino acids which have nitrogen in their ring structure are called as heterocyclic amino acids. For example, histidine and proline. A particular property of amino acids is the ionizable nature of the amino and carboxylic groups. Therefore, at a certain pH called as the isoelectric point, the amino acid can be both positively as well as negatively charged, so that the net charge cancels out and becomes 0. At a pH lower than this isoelectric point, the amino acid behaves like a cation, and above this isoelectric point, it acts as an anion. These amino acids undergo condensation reactions to form peptides. The bond formed between two amino acids after a condensation process is known as the amide bond or the peptide bond. So, peptides are actually nothing but chains of amino acids. If a peptide contains two amino acids, it is known as a dipeptide, and if there are 3 acids in the peptide, it is called a tripeptide. If there are a few amino acids, it is known as an oligopeptide, and if a large number of amino acids are present, it is known as a polypeptide. Now, proteins are macromolecules which are polymers of amino acids. There are 21 different types of amino acids, therefore, a protein is a heteropolymer. Proteins are the building blocks of an organism, and each protein has a specific sequence of amino acids. Now, certain amino acids are essential for our health and they have to be supplied through our diet. Hence, dietary proteins are the source of essential amino acids. Therefore, amino acids can be essential or non-essential. The non-essential amino acids are those which our body can make, while we get the amino acids which are essential from our food. Friends, let us now discuss the structure of proteins. There are 4 levels of organization of proteins. These are the primary structure, the secondary structure, the tertiary structure and the quaternary structure. The primary structure of a protein is mainly the sequence of amino acids in the polypeptide chain, which is held together by peptide bonds. The sequence of amino acids, that is, which is the first amino acid, which is the second amino acid and so on, is known as the primary structure of a protein. So, a protein is actually imagined as a line. The left end is represented by the first amino acid and the right end is represented by the last amino acid. The first amino acid is also called the N-terminal amino acid and the last amino acid is known as the C-terminal amino acid. So, a protein thread does not exist throughout as an extended rigid rod. Instead, the thread is folded into secondary structure which comprises of an alpha helix and beta sheets. When the primary structure is folded like a revolving staircase, it forms the secondary structure which is known as the alpha structure. In proteins, only right-handed helices are observed. The other type of secondary structures are the beta-pleated sheets. Beta-pleated sheets are formed when two or more polypeptide chains are lined up side by side. In that case, each individual segment is known as the beta strand. Unlike the alpha helices, which are coiled, beta strands are fully extended structures. Now, the long protein chain can also fold upon itself like a hollow woolen ball, giving rise to the tertiary structure. This gives us a three-dimensional view of a protein. Tertiary structure is absolutely necessary for many biological activities of proteins such as globular proteins. These structures form due to interactions between the side chains of the primary structure. Some proteins are an assembly of more than one polypeptide or subunits. For example, linear string of spheres or spheres arranged one upon each other in the form of a cube or a plate gives rise to a quaternary structure of a protein. Example is adult human hemoglobin. Hemoglobin consists of 4 subunits. It has 2 alpha subunits and 2 beta subunits. Now, proteins can carry out a variety of functions in living organisms. Some transport nutrients across the cell membrane, some fight infectious organisms, some are hormones and some are enzymes. Collagen is the most abundant protein in the animal world, and ribulose bisphosphate carboxylase oxygenase, which we know as Rubisco, is the most abundant protein in the whole of the biosphere. Then proteins may also be structural proteins like keratin, which is present in our hair, or contractile proteins like actin and myosin, which are found in muscle. Some may also act as storage or reserve proteins. For example, ferritin protein is responsible for storage of iron. Then there are globulins, which are associated with providing protection against foreign particles in the form of antibodies. Some proteins like rhodopsin act as receptors to various stimuli and help in performing downstream signaling. Now friends, we shall discuss about the other biomolecule known as carbohydrates. So the acid insoluble pellet that we had also has carbohydrates as another class of biomolecules. Carbohydrates are aldehydes or ketones containing multiple hydroxyl or OH groups. They are classified as monosaccharides, oligosaccharides and polysaccharides. Now monosaccharides are simple sugars that cannot be broken down further into more simple forms. For example, glucose. Depending on the number of carbon atoms, monosaccharides can be of various types. For example, trioses have 3 carbon atoms like glyceraldehyde, tetroses have 4 carbon atoms like erythroses, pentoses have 5 carbon atoms, for example ribose, hexoses have 6 carbons, for example glucose, fructose, galactose and mannose, and heptoses have 7 carbon atoms, for example pseudoheptulose. Oligosaccharides are composed of 2 to 9 monosaccharides. Depending on the number of carbon atoms, oligosaccharides can be disaccharide or trisaccharide or tetrasaccharide, etc. Disaccharides are the simplest oligosaccharides that are made of 2 monosaccharides joined together by glycosidic bonds. For example, sucrose, maltose and lactose. Sugars can be reducing or non-reducing. Now what do we mean by reducing or non-reducing? Reducing sugars have a free aldehyde or ketone group, for example maltose, and non-reducing sugars do not have this free aldehyde or ketone group, and the example for this is sucrose. Now what are the functions of these simple sugars? Different sugars are involved in different processes that take place in our body or in the environment. For example, Glyceraldehyde and riboses are involved in respiratory and photosynthetic pathways, and glucose, which we all know, is the blood sugar of humans. Now let us move on to polysaccharides. Polysaccharides are made of hundreds and thousands of monosaccharides. So polysaccharides basically are long chains of sugar, just like a cotton thread containing different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharide consisting of repeating units of the monosaccharide glucose. In plant tissues, a variant of cellulose known as starch is present, which stores energy. Animals have another variant which is known as glycogen. In a polysaccharide chain like glycogen, which is a branched sugar, one end is reducing end and the other end is a non-reducing end. Now some polysaccharides form such complex structures that it actually determines the function of that polysaccharide. For example, starch forms helical secondary structures so that it can hold iodine in the molecules of the helical portion. Cellulose does not contain these complex helices and therefore it cannot hold iodine. Now plant cell walls are made of cellulose. Even paper made from plant pulp is cellulose and cotton fiber is also mainly cellulose. Now there are more complex polysaccharides present in nature that have amino sugars. These are chemically modified sugars such as N-acetyl glucosamine and N-acetyl galactosamine. Exoskeleton of arthropods are composed of a complex polysaccharide called chitin, which is a homopolysaccharide. Why homopolysaccharide? Because it contains repeating unit of just one monosaccharide, acetyl glucosamine. Now some of the complex polysaccharides are heteropolysaccharides, that is, they are composed of more than one kind of monosaccharides. For example, hyaluronic acid, which consists of two monosaccharides, glucuronic acid and N-acetyl glucosamine. Now the next type of biomolecule that we are going to talk about is lipids. Lipids are generally water insoluble molecules called fatty acids, which may be esterified to an alcohol group. A fatty acid has a carboxyl group attached to an R group. The R group is a hydrocarbon chain consisting of 1 to 19 carbons. For example, palmitic acid has 16 carbons, arachidonic acid has 20 carbons. Common examples of lipids are butter, ghee and cooking oil that we use every day. Fatty acids are of two types. What are these? These are the unsaturated and the saturated type of fatty acids. Now what is the difference between unsaturated and saturated fatty acids? Friends, saturated fatty acids do not have double bonds and they have straight chains. For example, palmitic acid and stearic acid. While the unsaturated fatty acids, they have a double bond in them which bends the hydrocarbon chain. For example, oleic acid, linoleic acid and linolenic acid. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids. Here the fatty acids are found esterified with the glycerol. They can be seen as monoglycerides, diglycerides and triglycerides. So in monoglycerides, as the name suggests, one molecule of fatty acid is esterified to a glycerol molecule. In disaccharide, 2 molecules of fatty acids, and in triglycerides, 3 molecules of fatty acids are esterified to glycerol. These are also called fats and oils based on their melting points. Oils have a lower melting point as they have unsaturations due to which they remain liquid even in winter. Now in some triglycerides, one fatty acid may be replaced by a phosphoric acid, due to which they are known as phospholipids. The phosphoric acid may be linked to other groups such as choline in lecithin. Now some tissues, especially the neural tissues, have lipids with more complex structures. In sphingophospholipid, a sphingosine group is present instead of glycerol. For example, sphingomyelin, it is a type of sphingophospholipid which is a major constituent of nervous tissues. Then friends, you must have heard about cholesterol. Cholesterol is a complex lipid that contains 4 fused rings instead of straight chains. It acts as a precursor for formation of vitamin D and steroid hormones. Cholesterol is a constituent of cell membrane and helps in maintaining the fluid structure of the membrane. The fourth biomolecule that we are going to discuss is nucleic acids. Nucleic acids are long chains of polymers of repeating units known as nucleotides. There are 3 components of a nucleotide: a nitrogenous base, a sugar and a phosphate group. The nitrogenous bases are cyclic aromatic molecules that are insoluble in water. There are 2 types of nitrogenous bases, which are known as purines and pyrimidines. The purines found in DNA and RNA are adenine and guanine, which are also denoted as A and G. The pyrimidines found in DNA are thymine and cytosine, whereas uracil is found in RNA instead of thymine. The sugars found in nucleic acids are 2 types of pentose sugars, which are known as ribose and deoxyribose sugars. Ribose sugars have a hydroxyl group at the 2' end and are found in RNA, whereas deoxyribose sugars lack the hydroxyl group at 2' end and are found in DNA. Now, the sugars and nitrogenous bases that we have just discussed associate to form a nucleoside. When a nucleoside is associated with a phosphate group by the process of esterification, it is known as a nucleotide. Adenosine, guanosine, thymidine, uridine and cytidine are nucleosides, whereas adenylic acid, thymidyllic acid, guanylic acid, uridylic acid and cytidylic acids are nucleotides. Nucleic acids are formed by condensation of nucleotides and formation of phosphodiester bonds, which we will discuss later. Now, there are 2 types of nucleic acids. Can you name those? They are the DNA and RNA, the full forms of which are deoxyribonucleic acid for DNA and ribonucleic acid for RNA. Now, friends, let us talk about DNA first. The DNA consists of 2 chains of deoxyribonucleotides that run anti-parallel and are complementary to each other. Now, what do you understand by complementarity? There is actually a base pairing between the 2 strands of DNA, which is responsible for their secondary structure. Now, the secondary structure which is exhibited by DNA is the famous Watson and Crick model. In this model, the 2 strands are held together by hydrogen bonds in between the base pairs. Now, the question arises, which 2 bases will pair with each other? Adenine, which is represented as A, pairs with thymine or T, while guanine forms a pair with cytosine, that is pairing between A and T and between G and C. Now, 2 hydrogen bonds develop between A and T and 3 hydrogens form between G and C. This is why the GC pair is stronger than the AT pair. The helical structure of DNA is coiled upon itself and gives an appearance of a spiral staircase. The backbone of each chain is formed by alternate sugar and phosphate residues. The nitrogen bases are projected more or less perpendicular to this backbone, but they face inside. Now, each step of the staircase is represented by a pair of bases. At each step of ascent, the strand turns by 36 degrees. Now, one full turn of the helical strand would involve 10 steps or 10 base pairs. So, if we try to draw a straight line through the helix, the pitch or distance in one complete turn would be 34 angstrom and the rise per base pair would be 3.4 angstrom. We can actually calculate the pitch ourselves. If we say there are 10 base pairs in every turn and each turn covers a distance of 3.4 angstrom. So, 10 bases or one complete turn, which is called a helix pitch, would cover 10 multiplied by 3.4 angstrom, which is 34 angstrom. This form of DNA that we have just discussed with all these salient features is called the bDNA. There are several more forms of DNA about which you are going to study in higher classes. Now, DNA, friends, is the genetic material of organisms, and it is the basis of heredity, which causes transfer of information from one generation to the next. Now, moving on to RNA or ribonucleic acid, it is a single chain formed by condensation of ribonucleotides. The 4 nitrogenous bases in RNA are adenine, guanine, cytosine and uracil instead of thymine. There are different types of RNAs, for example, messenger RNA, ribosomal RNA, transfer RNA, etc., which perform diverse functions about which we will study later. So, friends, today we have talked about the 4 different types of biomolecules that are present in humans. Can we name these biomolecules together? These are amino acids that form proteins, carbohydrates, lipids and nucleic acids. In the next lecture, we shall discuss about the different bonds that are present in these biomolecules, metabolism and the role of enzymes in the metabolic reactions in body. Thank you.